Thank you for your interest in CultureTrax. This video explains why we developed CultureTrax and then gives you a brief tour of the platform. It's about 20 minutes long. If you would like to skip directly to the software demonstration section, that starts at the 6-minute 45-second mark.
As we all know, over the last 20 years, stem cell science has become enormously complex. Experiments take a long time and involve collaborations between many people and even different laboratories. In addition, data sets that we generate are large and complex because we're using the most advanced types of analytical technology for genomics, molecular analysis, imaging, and even linking to patient health records. This massive amount of data being collected in stem cell work really does present a data management issue. About 4 years ago, we began to look into this challenge. We realized that although there is a huge data management issue, virtually all stem cell scientists we talk to are using classical data management methods centered around the paper lab notebook. In addition, methods for capturing and sharing protocols are fairly primitive and really not up to the challenges of 21st century science. So we set out to create a domain-specific electronic lab notebook for stem cell science. As we worked on this, however, we realized that the real need is for a common data language designed to facilitate the transfer of information between stem cell scientists and to make the process of doing the bench science itself more efficient. Many of the funding agencies, like the NIH in the United States, are focused now on data management. Last year, the NIH published for the first time an institute-wide data management strategy and created an entire office of data management. Much of this effort is centered around these FAIR data management principles, which arose from an international working group 4 to 5 years ago. The goal is to make data that is generated findable, accessible, interoperable, and reusable for both people and machines, because it is becoming increasingly important for machines to find the right data over the internet. To actually have fair, compliant data, there has to be some standardization of how people record and manage their data. In addition, it requires a semantic data language. Any given analytical result needs an experimental context that's understandable to the person looking at the data. This is how we now envision CultureTrax, a common standardized semantic data language for stem cell science. Here's a picture of how this concept might work in practice. In this scenario, imagine Lab A on the left is taking patient fibroblasts, converting them to iPSCs, characterizing them, and putting them into a cryopreserved stem cell bank. Along the way, they are linking the patient's medical data and doing whole genome sequencing on those cells. Then Lab B, maybe even a few years later, takes some cells out of those banks, expands them using one protocol, differentiates them into dopaminergic neurons and astrocytes, and puts those together into a disease model for Parkinson's. Along the way, they are generating a lot of microscopic images, and at the end, doing some drug testing and generating gigabytes of analytical results. A scientist looking at those results might see something interesting and want to check back and see what the genomic data and medical records have to tell them. In this scenario, because all of the steps in this entire process over several years across 2 labs are linked together through the common CultureTrax data language, they can quickly get back to the medical and genomic data appropriate for that specific patient sample, even though they were done in a different laboratory years before. Here is how the CultureTrax semantic data language for stem cell culture is actually structured. The basic building blocks are what we call actions. These are familiar procedures that you might carry out in cell culture, like feeding your cells, passaging, or running analytical methods. The materials that you use to carry out the actions are also data elements within the system, as are the methods that you use to carry out the actions. The actions, in turn, are used as building blocks to create a day-by-day protocol template. This is the actual procedure that you will use for your entire experiment. To actually carry out cell culture, a protocol template is selected and applied to a line of cells that you are going to culture. CultureTrax also tracks the culture process by the vessels and the wells within those vessels that contain the cells. All of these are places to attach data that you record as you actually run the experiment. So, for example, on day 3, I am actually going to be feeding my cells with E8. CultureTrax gives me the materials and methods I need to do that on that day, and then gives me a convenient place to record the lot numbers. I can also record observations and results and attach those to any of the data objects within this ongoing track of my culture. For example, here, when I'm ready to passage the cells on day 4, I've attached a photomicrograph and a recording of the confluence of the cells, indicating they were ready to passage. I can also, when I run the PCR assay in this case, attach the PCR results to that same day in the CultureTrax. Now let's look at the CultureTrax platform itself. CultureTrax is cloud-based. You access it through a secure URL for your lab group on a web browser on any computer or mobile device. Here is the view of my culture work for today. As you can see, I have 3 culture tracks going on. 2 of them are patient iPSC lines that I'm expanding and QCing using this iPSC expand and QC protocol template. We see the cell line names here, and CultureTrax is keeping track of the passage number for each of these cell lines. Let's look at the first cell line. My plan for today is simply to feed these cells using E8. If I go into the details of that action and look at the materials, here I have my E8 material listed. I can put in a batch number and record the amount in mils per well that I feed the cells with. If I save those action details, I'm really basically done recording that work for today. If I check the work as complete and check the culture track complete, I'm done. Now let's look at our second culture track. If I look at the notes for this culture track, I took a photomicrograph when I came in this morning and determined that the cells were at about 80% confluence, certainly ready to passage. So what I want to do is take 4 of the wells of the 6-well plate and split them 1 to 6 so I can continue expanding the cells. The remaining 2 wells I'm just going to feed with E8 to keep as a backup in case something goes wrong. The first thing I need to do to set up the passage action is to let CultureTrax know what vessels I'm going to put these cells into. A single well of a 6-well plate in a 1 to 6 expansion roughly corresponds to a 100 millimeter dish. And since I'm passaging 4 wells, I'm going to passage into 4 of these 100 millimeter dishes. I create those vessels, and there they are. Now if I look at the materials for this action, I remember that half millimolar EDTA reagent is out in the lab. With a single click, I can open the recipe and get all of the ingredients, the amounts, and the full prep procedure for this reagent so I can make it if needed. Once I save my action details, there we go. Now, one of the things, because this is more complex, I may want to actually look at the vessels that I have created to make sure I'm doing the right thing. Here you can see that we started the day with a single 6-well plate, and we're ending the day with 2 of the wells from that plate in my current culture track at passage 6. And the 4 new 100mm dishes are in a new culture track at passage 7. CultureTrax has automatically incremented the passage number. So once I click that those are done, I've completed recording my work for the day. Now let's look at the third culture track. Here I'm doing a different protocol template, a cardiomyocyte differentiation for a drug test. I'm starting the day with 3 24-well plates, and I'm going to be performing, well, let's see. I'm going to be performing an endpoint assay from 1 well of each of the plates using qPCR to make sure that these cells are properly differentiated. The remaining wells are just going to be fed in the cardiomyocyte differentiation medium. Let's look at the assay action. The materials for this are just a TACMan assay kit. What's interesting here, though, is the notes and data. Attached to this action, I've put a link in to qPCR results. And if I open that link, it takes me to a spreadsheet with all of my qPCR results, automatically tied into the action in the sequence of my culture track. At this point, you may well be wondering how CultureTrax actually knows all this detailed information about every single step in my experiment. The key is the protocol template. If I look at my dashboard, I can see I have libraries of protocol templates, documents, and materials, all of which can be reused for multiple experiments. If I go to the protocol template library, I can see that I have protocol templates for the entire lab. I can even keep personal ones that I may be working on or are not quite ready to share. I also can look at protocol templates that have been shared with me or that I've shared with scientists in other laboratories. These are sorted by type, and I can search for them using some powerful search tools, but it turns out this one that we want to look at is right at the top of the list. So let's open that up. I'm going to put it in editing mode because we may want to make a few changes to this. You can see, in addition to naming it, I can link documents that I need to carry out the protocol template or that were really the source of the material. The heart of the protocol template, however, is the action timeline. Here I array all of the actions that I want to carry out for this experiment on a day-by-day timeline. Here, for example, is an EDTA passage that I used to start this cardiomyocyte differentiation. All of these actions themselves are fully editable. The materials and methods that I need and documents that I might want to look at when carrying out the action. To give an example of how you might want to edit this, suppose I discover and validate in the lab that instead of using these expensive, ready-to-use CultureTrax plates, I can use lab-made Matrigel plates. In order to make that change in the protocol, I simply remove the CultureTrax plate and go to the vessel material type. If I click on Add, it takes me to my material library where all of the materials that we use in the lab for cell culture are stored. If I do a quick search, I quickly turn up the Matrigel coated plates. If I select those, they're immediately added to the passage action in this particular protocol template. In addition to editing and creating protocol templates, there are a few more things I can do with them as well. One is I can share them with colleagues in other laboratories. Those colleagues, of course, have to be users of the CultureTrax platform, but if I type in their login email addresses, I can send the protocol template along with all of its linked materials to their library so that they can use it in their laboratory. In addition, I can generate a written report of the protocol template. This is in the form of a zip file which contains all of the documents that I've linked into the protocol template, as well as an editable Word file which has every detail of this protocol template in a human readable form. Every material recipe, all of the actions, and the timeline. Speaking of sharing, there's one more element of CultureTrax I'd like to show you. Here we are back on my culture work for today, almost complete. One aspect of CultureTrax, however, is that I can share that work with others. In fact, all three of these CultureTrax have been shared. In addition to my own work, I can also look at other people's work that they have shared with me. For example, let's take a look at the CultureTrax that Tori has shared with me. And here we go. I can do work on these CultureTrax and it'll automatically be recorded that way in the system so that Tori has an accurate record of what I did. So that gives you a good idea of the current CultureTrax platform and how it might help you plan, record, and document your work in the lab. As with any software platform, we're always making new developments and adding new features, and there's some really important ones that we're planning to add over the next several months. Let me give you a sense of what those will look like. The most important new feature is this new culture work overview shown here in an early design wireframe. The idea is instead of looking at a single day's worth of culture work at a time, we actually can see an entire week at a glance. This way we can see everything that I have planned for today in the context of the work that I've just completed and in the context of the plan for the following days. Another important feature we're working on is greatly enhancing the search and reporting functionality in the platform so that you can quickly and easily find the data you're looking for from the work you've done in the past based on a variety of different search parameters. Moving forward, we have a roadmap for the following 2 years in developing new features and capabilities for the platform. These include things like being able to manage the cryovials in your liquid nitrogen tank, searching for them based on any characteristic of the culture work that was done to create them, not just the things you happen to record. We're also looking at things like being able to access the platform efficiently and input or receive data using a smartphone, which many of our bench scientist users have asked for. Finally, we're adding a number of tools to support enhanced collaboration between scientists, things like interlab data sharing and integration with other platforms such as existing electronic lab notebook systems. Our goal is to make CultureTrax work for all of the different people in the lab. A big focus for us has been bench scientists, the ones who actually go in and do the work every day. Here, the focus is primarily on efficiency, being able to plan, document, and record all of your work, find the results quickly and efficiently. But in addition, of course, we have to satisfy the needs of the principal investigators and lab managers. We're more concerned, typically, about getting an overview of things happening in the lab and standardizing what happens so that the work is reproducible and has high impact. CultureTrax is currently in beta testing, and the way you work with us at this point in time is through our Early Access Partner Program. In this program, we're forming strategic partnerships with key thought leader laboratories, providing them with early access to the CultureTrax platform prior to when we release it commercially. What's in it for our EAPs? One, of course, is getting this enabling tool for stem cell research available as soon as possible. You'll also have extraordinary influence on the development of the coming product features, and of course we'll give you priority training and support. We hope that you'll consider joining the 9 current EAP laboratories in 6 different institutions that we have today.
For more information on getting started with CultureTrax, please contact us.